Hi, everyone. I'm really excited to be participating in this conference today. I'm Susan Catalano, and I'm going to speak to you today about the small molecule oligomer receptor antagonists that our company has discovered and is forwarding to the clinic for disease-modifying treatments as Alzheimer's therapeutics. So I wanted to start by thanking the individuals who are responsible for this work, and you can see some of their names on this slide, as well as our collaborators. Tara Spires-Jones from Brad Hyman's lab, Tavio Orancho, as well as Dr. Mehrdad Shamlou from the Stanford Neurobehavioral Facility, and the folks at JSW, now QPS Pharma. These are really the individuals who've been behind us since the beginning, and it's been really wonderful working with them. So the first slide presents some of the evidence from the literature that Abeta oligomers actually are one of the major culprits behind Alzheimer's disease. They certainly accumulate with age and produce reduction in human cognitive ability, and I think we've seen some recent publications that actually add to that evidence. And they produce a transient synaptic regression and deficits in not only surface receptor expression, but also synaptic plasticity. And they are therapeutics that are directed against them can actually prevent and reverse the pathological effects that we see in disease models. Certainly apologies to anyone whose work I've left off of this slide. There really is quite a large body of evidence that suggests that these are toxic species that are producing the synaptotoxicity we see in the disease. There are some issues currently with the Abeta oligomer hypothesis for Alzheimer's disease, and I think these have been raised in some recent reviews in the literature. Certainly it's controversial, the exact oligomer species that's responsible for the synaptotoxicity that we see in the disease is yet to be identified. But from our standpoint, certainly the concept that there is a single species, a single toxic species, is not necessarily the case. We may be dealing with ligand that essentially has structural heterogeneity, and we're simply going to have to come to terms with that. So it's a fundamentally different view of how the disease takes place. And I think one of the things that's guided us as we've engaged in our discovery process is the possibility that there's actually mass action behind this phenomenon, that the Abeta oligomers behave as pharmacological ligands and exhibit saturable and specific binding to the neurons in the central nervous system. And certainly if it's true that there is a pathological ligand that's interacting with specific receptors in a ligand receptor interaction, that would be good news for the patients, since generally, industry is very familiar with how to solve those kinds of problems. So if they are specific ligands, then effective anti-oligomer small molecules can actually tell you both what the identity of the molecular interactions downstream of that might be, and they can also represent effective therapeutics, since they would actually be interfering in this fundamental process. So the strategy that we took as a company was to find effective anti-oligomer drugs, behaviorally efficacious drugs, and then figure out how they interacted with the signaling molecules to agonize or antagonize them as we worked backwards into the mechanism of action. So there were a couple of principles that we engaged in early on to guide our efforts, and some of those are listed here. And when we set out, we actually wanted to use only a very novel drug-like library. If you're going to engage in very difficult screens, as you'll see in the rest of the slides, it really is incumbent on you to use very high-quality starting material. And then we focused exclusively on assay systems that really get as close as possible to modeling mature nervous system function. And for us, that was hippocampal and cortical neurons that are a minimum of 3 weeks in vitro. At that point, they really are at the stage where they're actually modeling a connected nervous system. And then most fundamental of all, I think, is that we knew that we weren't going to actually be able to shed any light on the Whichmer debate. So we decided not to decide a priori which oligomer is the villain, or even that there would be a single villain, but to use all of the forms of oligomeric Abeta that we can get our hands on. And certainly, if there's some kind of epitope common to all of them, the same drug would be effective. So initially, when we set out, you could use a small molecule library. There are many libraries that are commercially available that are CNS drug-like libraries. Most of them are based on scaffolds of existing therapeutics. And we were pretty convinced that that wasn't going to provide us with a fundamentally disease-modifying candidate. So what we did was generate our own proprietary library. And one of the co-founders of our company, Gil Richton, was the medicinal chemist who came up with the process for our library generation. And so what he did was produce a library that takes its point of departure from natural products, even though these are not natural product libraries. What Gil does in this process is remove all of the reactive chemistry from natural product starting material, so that at the end of it, you're left with CNS drug-like compounds that hopefully preserve the biological diversity and the novelty that's contained in the original starting material, but removes all of the reactive chemistry that creates artifact in all kinds of assay systems. And I can't stress that point enough, that reactive chemistry really is something that's really doesn't help you find out or investigate biological phenomena, and really doesn't play much of a role at all when you're investigating those kinds of phenomena. Certainly not when you're looking for therapeutics either. So that's the basis of our screening library. And then the basis of the assay system is shown here in this slide. Mature primary rat hippocampal cortical cells in vitro, minimum of 21 days in vitro. By that time, they are connected with each other. They're expressing the full complement of their synaptic proteins. They exhibit oscillatory electrical signaling with each other at that point, and serve as the most elemental basis for investigating mature nervous system function. And then to those cultures, we actually add various oligomeric preparations. And as I said at the outset, we use them all. We've used all of them to calibrate our screening systems and these assay systems. And I'm going to take you through two of them right now. The first is probably in the most widespread use in the academic community, and that would be oligomers that are made from synthetic Abeta, synthetic human Abeta 1 to 42. And the key for us here has been to characterize these preparations using a very wide variety of methods, everything from MALDI-TOF to two-site ELISAs to immunocytochemistry, and most importantly, I think, non-denaturing gel conditions or native gel conditions. And it's been challenging trying to get those methods calibrated with respect to some of the more commonly used methods. But we were convinced that we really needed to quantify these oligomers in a way that wasn't going to introduce artifact into the system. So what you can see here in figure in the image mark A is 3 lanes from native gel, in which we've, lane 1 contains the 1 to 42 Abeta in freshly prepared monomeric solution, and the oligomers that have been allowed to oligomerize for 24 hours are shown in lane 2. And then for comparison, a freshly prepared Abeta 1 to 40 solution that's predominantly monomer is shown there. Monomer runs at a higher molecular weight under these conditions than you would expect from the globular size standards, and so we've marked it with an arrow. And what you can see is that in the oligomeric situation, we have a range of molecular weight sizes, and we also have a fair bit of monomer, and this is something that we think is probably underappreciated. The range of oligomeric peaks can also be seen in the MALDI graphs that are shown there on the right side of the slide, compared to the vehicle, which is the neurobasal media with the B27 supplement that's shown in the lower right. And in both cases, you can see the albumin peak there at about 66 kilodaltons, and some media proteins down at lower molecular weight sizes. So you have a range of oligomers that we can see in both the native gels and the MALDI, as well as a peak, a monomer peak, where you would expect it in the MALDI of 0.5 kilodaltons. So these synthetic preparations are a mixture of high molecular weight oligomers and a fair concentration of monomer. In contrast, the material that we isolate from human Alzheimer's disease patients is shown in the next slide here. This material, again, we're looking at the native gel conditions in A, and B through E are the MALDI-TOF of the individual patients, 2 Alzheimer's patients shown in the upper panels and 2 age-matched normal individuals in the lower 2 panels, C and E, as well as the 2-site binding ELISA that's shown in the table on the lower left. And what you can see when you look at the native gels is, again, no 2 patients are actually showing complete registration in the species there. They are higher molecular weight, and what's really interesting is that we really can't see much in the way of monomeric Abeta under these conditions, and that's verified by the MALDI records themselves. You have this range of sizes that you can see on MALDI in the Alzheimer's patients that really are absent in the normal controls. The relative lack of monomeric Abeta is, can be seen with the ELISAs, where this is the 2-site binding ELISA that was pioneered by Harry Levine. That's also been published on from the Selthorne Holtzman labs as well. This basically uses either identical capture and detection antibodies or distinct capture and detection antibodies to quantify the relative amounts of oligomer and monomer that are contained within a given preparation. And in contrast to the synthetic Abeta 1 to 42 oligomers that are shown in the central panel, where you do have a fair concentration of monomer, about 1/3 monomer. In the human material, you really don't have detectable levels of that at all, and that certainly was surprising to us, in addition to the heterogeneity that we saw between the human samples. So certainly very different features, some overlapping between the synthetic and the human oligomers. We've used other preparations as well, and we compare all of them side by side in these assays that we use to screen to find the successful drugs. So one such assay is shown in this slide. This is an assay that actually measures membrane trafficking rate. What you're looking at here is a cargo dye that actually loads into the vesicles in endocytosis within the cell. And that's shown in the image labeled vehicle, where you're looking at neurons and glia that are completely filled with these purple vesicles. And over time, what happens is the vesicles are trafficked and their contents are released to the outside in exocytosis. And when that happens, the cargo dye actually precipitates as a needle-shaped crystal on the surface of the cells. And you can see that in the center picture. And what several labs have published previously is that Abeta oligomer exposure actually accelerates the rate of membrane trafficking. In this case, it accelerates exocytosis selectively. And so a given culture that's treated with vehicle and Abeta has the appearance that you can see in these 2 images labeled respectively here. And so what the end game is really when you're looking for effective therapeutics is to find those small molecules that actually are able to restore a membrane trafficking rate back up to normal levels in the presence of Abeta oligomers. And an example of that would be shown in the case of the anti-Abeta antibody. That's 6010, and when we apply it at successively higher concentrations, we see it very effectively take out the Abeta that's there in culture. And that's represented in the graph on the right-hand side, where the y-axis is actually representing the amount of cargo dye contained in the vesicular compartment, expressed as a percentage of vehicle, and the x-axis is showing the concentration of total Abeta, a subset of which is oligomers. So we chose this assay because memory trafficking rate in general are critical for normal synaptic plasticity processes. The insertion and withdrawal of AMPA receptors is critically dependent on it, for example, as well as many of the other basic cellular processes. So this kind of an assay is a very large downstream basket that's going to catch a variety of mechanisms whereby Abeta oligomers can impact the cells. What you can see in this slide is a side-by-side comparison of various oligomeric preps compared, both the human and the synthetic that I showed you previously, as well as some semi-synthetic preparations that we've used. And again, the axes are the same as previously, with the y-axis being a reflection of the amount of cargo dye that's in the vesicular compartment. One thing that leaps out at you immediately is that there's quite a wide concentration range to these effects, but that all of these oligomeric preps are in fact active. And I think that was the activity, the broad neuroactivity of multiple preparations of Abeta oligomers, something that's been published previously by Jim Cleary and others. It's really just a question of potency. So what you can see is the amount of time, not only the EC50 for all of these preparations at causing membrane trafficking rate deficits in vitro in these cultures, but also the amount of time that it takes for these preparations to reach their maximum impact on membrane trafficking rate. And you can see we have quite a range of concentrations. So the important thing is to recognize that certainly the top end of these curves are marked by rapid toxicity, and our goal as a company was to stay very far away from frank cell death phenomenon, really interested more in interfering in the plasticity deficits that are a feature of sublethal doses of Abeta. And so that's what's used exclusively in the assay systems, is the lower concentrations of Abeta that are not going to cause the rapid cell death that we see at higher concentrations. So many of the images that I'm going to be showing you throughout this presentation are based on the synthetic oligomer preparations. These are the least potent preparations that contain the largest or highest concentration of monomer. I think the reason for this is that this is probably the preparation that's in the most widespread use within the community, research community. And so an example of that, what we see when we look at the localization of that kind of material in these cultures is shown on this slide. And what you're looking at are cells in culture where the blue is showing that's DAPI stained nuclei, the green would be MAP2, and the red would be the Abeta oligomer localization. And when you look just at the one color image of Abeta oligomers in the right-hand panel, what you can see is sort of two varieties of Abeta localization. One is this punctate staining that is at the synapses in one particular neuron, but not all of the neurons in culture. I think this was originally shown beautifully by Bill Klein and Grant Kraft and their collaborators. The central feature of oligomers that certainly first captured our imagination a while ago was that you had some neurons that were completely decorated with oligomeric species and others right next to it that had no binding whatsoever. You also see the cell bodies, both neurons and glia, having various amounts of Abeta positivity to them. And this is something that we see, and some of that's quantified in the next slide. And so what we did here was use actual image processing to quantify the amount and intensity of the Abeta localized not only within the synaptic puncta, but also within the cell body. And so what you're looking at in images A and B are the Abeta oligomers that have been exposed to the cultures for about an hour. And then in the middle panel, we're looking at a freshly prepared monomeric solution of Abeta oligomers that have been applied as well. And then in the bottom two panels, G and H, is oligomers that have been added in the presence of 6E10 antibody. And the three color images showing you everything that I mentioned previously, and the black and white image, just the Abeta itself. And what we then did was quantify the amount of Abeta intensity that we saw in the synaptic puncta of neurons and glia, and that's quantified in the graph on the upper right, with the neurons being the solid red and the glia being the open circles, the open squares there. And what you can see is that the Abeta that's localized to the synaptic puncta actually fits a single site binding curve with a KD in 480 nanomolar range. Whereas the Abeta that's bound to the glial cell bodies, which is shown with the open circles, actually exhibits two-site binding, a high affinity site that's again around 460 nanomolar, and a much lower affinity site that's somewhere in the micromolar range, high micromolar range. And when we look at the monomer binding, we essentially find that when fresh Abeta monomer is applied to these cultures, it does not exhibit any kind of saturable binding to either neurons or glia. So this is essentially an non-saturable binding phenomena that we're looking at. And the concentration ranges that we're working at when we look for effective anti-oligomer therapeutics is shown in the blow-up of the left-hand side of that graph that's shown underneath it. Again, focusing on concentrations of Abeta that are somewhere near the KD that we've measured at the synaptic puncta in the triple-digit nanomolar range. We've also done some comparison with 1 to 40 oligomers. And what you can see is that oligomers produced from 1 to 40, not only are they take quite a lot longer to actually form, but they're vastly less potent than the 1 to 42 oligomers, both in the binding assay that we measure, where the intensity of the binding is measured on the y-axis as a function of Abeta concentration, and also in the membrane trafficking assay, and that's shown in B. And we've also taken a look at scrambled Abeta, and that's not active in the membrane trafficking assay either. So by screening the library, we're actually able to identify therapeutics that are effective anti-oligomer therapeutics. And one of the features we were looking for were small molecules that were effective regardless of the order of addition. For example, in the membrane trafficking assay, you can see the photomicrographs of the actual phenotype compared side by side at the top. And in the bottom, you're looking at two graphs on the left-hand side, a situation where compounds are added before the oligomers. And on the right-hand side, what we refer to as a treatment mode of the assay, where the compounds are actually added after the oligomers. We identified molecules as hits only if they were able to be active in both situations. Again, this would imply a pharmacological mechanism of action to both Abeta oligomers themselves, as well as to the therapeutics. And then we also took a look using this membrane trafficking assay at whether we actually had a competition situation between the oligomers and the compounds for access to targets that are mediating membrane trafficking. And what you can see in the upper panel of graphs is, again, the same membrane trafficking assay. And here we're looking at three different small molecules in A, B, and C that are dosed against the least potent oligomeric prep, the synthetic Abeta oligomer prep. And what you can see is that as we dose the Abeta, which is shown in the red line in these graphs, against ascending concentrations of compound, we get a rightward shift that fits a shield of 1 and indicates that there's true pharmacological competition going on in this situation. The bottom set of panels, we've dosed these same compounds against oligomers that we've derived from human Alzheimer's patients' brains. And you can see the same kind of shift there. So what this is telling us is that we have a pharmacological basis to the mechanism of action of the oligomers and the compounds, and that the drug candidates are effectively making Abeta oligomers less potent and therefore less toxic. So we then took a look at these same candidates in the oligomer binding assay. What you're looking at in the photomicrographs is the amount of Abeta bound in the large squares, and then right next to that we're showing you the right-hand half of each of those images with the NAP2 staining, so that you can see the relative density of the neurons in these cultures and that they're comparable in each case. And again, in the Abeta oligomer treated panel on the upper left-hand side, you can see the oligomer is binding completely to a subset of the neurons that are contained there in culture. But in the presence of compound, we have a very profound loss of binding. In this situation, the compounds were added prior to the oligomers, and you can see the dose-dependent loss of oligomer binding quantified in the graphs on the right-hand side for 2 of these candidates that are able at higher concentrations to block up to 90% of the oligomer binding. We also did the same experiment in treatment mode that's shown in this slide. In this case, the Abeta is actually added before, excuse me. The compounds are on for an hour before the Abeta is added. And in this case, we actually have a true displacement of Abeta oligomer binding that's also dose-dependent and also reaches quite high levels of displacement of Abeta oligomers. This has never been shown by any other therapeutic, small molecule therapeutic that we're aware of. And it implies that the compounds are going to continue to be effective as the concentrations of oligomers rise throughout the disease. And so that's all well and good in culture, but what about the patients, which is certainly something that was the focus of our attention. And so to get at that question, we actually took advantage of the results that have been published by Tara Spires-Jones and Brad Hyman, in which they had described a halo, which in human Alzheimer's patients, tissue sections is actually 2 microns in width around the outside of a compact plaque that contains a particularly high concentration of oligomers. And what we did was actually use automated image processing to measure the intensity of the oligomers within that region around the outside of a compact plaque. And some examples are shown here where we have a thioS labeled plaque labeled in green and also have the Abeta visualized in red. And you can see in the lower right-hand side this image processing mask that measures the Abeta intensity within that 2-micron halo around the outside. And so what we did was essentially the ex vivo binding experiment, in which we incubated these tissue sections with fixed volumes of ascending concentrations of compounds and looked to see if we could actually displace the endogenous human Abeta oligomers from this region within the tissue, and the results are shown on the next slide. We actually did see a dose-dependent displacement of Abeta from that region. And some of the images are shown there in the upper right-hand side, and a quantification of that in the graph in the lower right. This was a significant displacement of endogenous oligomers from human tissue. Again, we're unaware of any other therapeutics, small molecule therapeutics, that have demonstrated this ability. This was done under non-equilibrium conditions and actually raises the exciting possibility that these therapeutics could potentially be used to calculate an off rate of endogenous human oligomer binding from tissue. So that's certainly something that we plan to do in the future. And so this is probably as close as you can get to patients without actually conducting the human clinical trials. So we were quite excited about seeing these results. We also examined the ability of these same therapeutics to actually rescue synapse loss that's caused by the oligomers. And oligomers cause varying levels of synapse loss. In the case of, again, this least potent Abeta prep, the synthetic oligomer prep that contains a fair bit of monomer, we actually see about an 18% loss of synapses in vitro following exposure, and that's commensurate to the amount of synapse loss that we see in the hippocampus of MCI patients. And when we add the compounds that are shown in the solid bars here on the graph and use image processing for synaptophysin in green, you can see that we actually have the compounds are able to completely prevent this 18% loss of synapses that we see following oligomer exposure. Importantly, when we dose them all on their own, which is shown in the open bars on the graph, we actually do not see any change in synapse number. So this is, again, very important for the mechanism of action. We're not interested in compounds that are able to either reduce or enhance the number of synapses when they're added on their own. We're not interested in neuroactive compounds per se. We're really exclusively interested in compounds that are actually able to stop oligomer effects on the tissue. So we took advantage of a biochemical assay described by Harry Levine several years ago and looked at the ability of these compounds to interact directly with Abeta oligomers themselves, either by dissociating oligomers or preventing their formation in the first place. And all of the lead compounds that we've identified are actually not active in these biochemical assays. So it suggests that these compounds are not interacting directly with oligomers themselves, but rather subsequent work indicates that they're actually interacting with receptor that that the Abeta oligomers are binding with. So we took these compounds and we actually went straight to behavior to see if they were going to be behaviorally efficacious when administered to various models of Alzheimer's disease. And this slide shows work that was done in collaboration with Ottavio Rancho's lab at Columbia University. And Ottavio has a wonderful model in which Abeta oligomers are injected directly into the hippocampus. And then these animals are trained in the fear conditioning apparatus and then tested for their ability to recall 24 hours later. And under these conditions, a single injection of oligomers into the hippocampus produces a very reliable deficit in fear conditioning that's shown in the red bar compared to vehicle injections that are shown in the blue bar. And so what we did was administer our compounds 1 hour prior to the Abeta addition through the same intrahippocampal injection. And this is exactly the same conditions that we're using in the assays themselves. And under those conditions, both compound 109, shown in the black bar, and compound 93, shown in the solid gray bar, were actually able to completely prevent the Abeta-induced deficits in fear conditioning. We also injected these molecules by themselves in the absence of Abeta, and that's shown in the hatched bars on the right-hand side of the graph. And importantly, neither of these compounds produced any deficits in fear conditioning or enhancement in fear conditioning when they were added on their own. So these compounds, again, are not neuroactive. They do not interfere with normal learning and memory processes. They're selectively interfering with Abeta oligomeric signaling. These were wild-type male black 6 mice, 3 to 4 months of age. We also tested these compounds for their ability to improve cognitive deficits in transgenic animals that were aged transgenic animals. And so for this work, we went to the Swedish London transgenic model. This was a model that was characterized by JSW over in Austria, now QPS. And this model exhibits an age-dependent accumulation of various structural forms of Abeta and an age-dependent deficit in cognitive functioning. It was published in 2011. And this is exhibited by both genders compared to their vehicle-treated non-transgenic litter mice. So this was the system that we were able to access for testing the compounds. And the first such study is shown on this slide. Here what we did was use 8-month-old female Swedish London mice that's shown in the red bar and the red lines. And here we treated them with compound 109 at 0.1 or half a mg per kg per day for 4 weeks, and then tested them in the Morris water maze. And what you can see is that on the second day of testing, when you compare the red to the blue, the transgenic animals actually exhibited significant deficits in learning compared to their wild-type litter mates. And on that day of testing, a treatment with a higher dose of 109 at half a mg per kg was able to significantly improve the cognitive deficits at brain concentrations in the double-digit nanomolar range, with no adverse behavioral effects observed in these animals. So we repeated this study, and that's shown in this slide, with 9-month-old male Swedish London mice, and here they were treated for 4 to 6 weeks at 1 or 10 mg per kg per day. And here on the second and third day of testing, we actually saw a significant deficit in cognitive performance that was improved with the 1 mg per kg per day dose of the compound. Again, brain concentrations measured in the double-digit nanomolar range with no adverse behavioral effects observed. In a separate cohort from the same study, we looked at compound 93, and again, here with this compound, the higher dose, the 10 mg per kg per day amount, was able to significantly improve the deficits that we saw in these animals, in the transgenic animals at that age. So we conducted another study with a second generation of molecules from the 109 series. These are orally bioavailable molecules that were administered for 42 days to the same Swedish London line of transgenic mice. And here we were treating them at 30 mg per kg per day. And what we saw in the Morris water maze was that we were able to significantly improve the cognitive deficits measured in the probe trial in these animals. Here we were actually dosing the wild-type animals as well and saw no adverse behavioral events in this study either. And lastly, what we did was actually treat aged animals. There were 9-month-old male Swedish London mice, and when we started this study, we treated them for almost 6 months with 2 doses of compound 1344 or 1346 at 10 and 30 mg per kg per day. Both doses of compound 1344 were actually effective at improving the deficits in fear conditioning seen in the transgenic animals. And the higher dose of compound 1346 was able to do the same. And again, measured brain concentrations were as expected, and no adverse behavioral events were observed. So when we added up all of these studies together, a very interesting pattern came out, and that's shown on the table that is shown here on this slide. What you're looking at is the 4 compounds that I mentioned previously, and in the upper part of this table, you can see each of their doses, either at 1 and 10 or 10 and 30 mg per kg per day, and right underneath that is the measured brain concentration expressed in nanomolar units. And then underneath that is a plus or minus, depending on whether or not we saw statistically significant behavioral improvements in those animals. And underneath that, we actually measured the are actually measuring the compound affinity at the target receptor that we believe Abeta oligomers are binding to, expressed in nanomolar units, and then estimated the percent receptor occupancy based on the Ki and the measured brain concentrations in these animals. And what we saw was that as long as we had 80% receptor occupancy at the target by the compound, as long as the compound concentrations were high enough to achieve 80% receptor occupancy, we actually had behavioral efficacy. Lower than that, we did not. So we were very encouraged by this. Again, additional evidence that there's actually a pharmacological basis at work here. So when you add it all up together, we were very gratified to see that the evidence, the bulk of the evidence, really suggested that there was a pharmacological basis to the action of oligomers. We had saturable binding to synaptic sites at a single site on mature neurons. All of the different forms of oligomers that we actually tested cause membrane trafficking deficits, albeit at different potencies. The receptor antagonist drug candidates that we have, as well as antibodies that are selective for regions of this target receptor, could both prevent and competitively displace oligomer binding, both to the brain cells in vitro as well as to the human-derived material. They could prevent and reverse the effects of oligomers on membrane trafficking and synapse loss. And they could prevent oligomer-induced cognitive deficits in the wild-type Alzheimer's model and reverse the cognitive deficits in the age-transgenic Alzheimer's model, and sustain that improvement for more than 5 months, as long as their receptor occupancy was above 80%. So it certainly indicated to us that the different sources and the sizes of Abeta oligomers have a common downstream effect that can be reversed by the same molecules. It also suggested that these in vitro assays were able to predict the behavioral efficacy and had an excellent track record of doing so. And all told, indicated to us that the compounds would represent promising disease-modifying therapeutic drug candidates. So when we got the evidence for behavioral efficacy of these compounds, we actually took the compounds then to really standard industry counter-screening assays. And you could go to a number of organizations for this kind of work. We actually tested these behaviorally efficacious compounds at over 100 receptors, looked to see what they bound to or activated or inactivated with really high affinity, affinities that were commensurate with the measured brain concentrations that we saw. And from that standard industry counter screen, we saw that the lead series really only had high nanomolar affinity for a single receptor. And when we went out to the standard radioligand binding assays, not only in the tissue homogeneous, but also in human brain tissue, we saw that they had single or double-digit KIs at displacing standard radioligand binding from this receptor. So they certainly fit the bill for classical ligands at this receptor. And what you're looking at here is an image of the target receptor. We're currently in the process of submitting these results for publication, in which we're going to be talking about the identity of this receptor, the target receptor. What you're looking at here is immunocytochemistry, localizing this target receptor to postsynaptic density sites in close apposition to synaptophysin-positive sites. So it's certainly at the right place at the right time to be playing a role in Abeta oligomer pathology at the synapse. And here what we've done in this slide is actually knock down this receptor's expression using siRNA in these mature cultures. We actually used varying concentrations of siRNA to knock down the protein expression as visualized with immunocytochemistry. And then in those same cells, so you're looking at target receptor expression in the lower panel of photomicrographs. And then in those same cells, we looked for Abeta binding, again measuring the intensity of both with automated image processing. And the results are graphed over on the left, in which we saw a really high degree of correlation between the amount of receptor expression lost and the amount of Abeta ligand binding lost. We actually were able to see more than 90% of the Abeta oligomer binding being mediated by this particular receptor. So there have been a variety of receptors that Abeta oligomers have been thought to bind to, and certainly we think the situation is likely to be quite complex. The most that's I think ever been reported has been about 50% loss of oligomer binding, as compared to the current 90%, more than 90%. Oligomer binding loss. We think the situation is likely to be complicated, but our interpretation of these results is that Abeta oligomers bind either directly to this target receptor or to a protein that's quite tightly regulated by this receptor. And the evidence is consistent with both at this point in time. The action of our compounds is actually to act as an antagonist of these receptors. One of the classic agonist-induced results at this receptor is caspase activation. And so in that respect, our compounds are not behaving like agonists. They do not appear to cause caspase activation, but instead are actually able to block agonist-induced caspase activation. So the evidence is consistent with not only a high affinity binding to this receptor, but also behaving as antagonists of this receptor. And so putting it all together, it gives you a model in which Abeta oligomers are binding to what's probably a complex of proteins, including this particular receptor, and in binding are actually able to induce signaling deficits that certainly include membrane trafficking deficits, ultimately resulting in synapse loss, receptor withdrawal from the plasma membrane, and synapse loss, and ultimately manifesting as memory loss. What our compounds are able to do is actually allosterically modulate this receptor and competitively displace the Abeta oligomer binding, and in so doing restore both the memory trafficking as well as the synapse number and the cognitive functioning back up to normal. And so we've sort of set this in motion in an animation that I'm going to ask Don to show you in a second. This actually is an animation that uses the atomic resolution PDB models for both this receptor as well as the oligomers themselves, which we graciously received from Dr. Vijay Pandey, who published the MR solution structure of the oligomers. What you're going to see is the N-termini, the disordered N-termini of the oligomer sort of waving around the core hydrophobic domain of the oligomer. And you're going to see the oligomers binding to this particular receptor. Again, the situation is likely to be a little bit more complex than what's depicted here, but for the purposes of animation clarity, we sort of simplified it. And you're going to see our small molecule in blue binding to an allosteric site on that same receptor and actually resulting in the injection of oligomers and the restoration of the signaling to normal. It's got a little bit of a soundtrack just so that you don't think your sound is malfunctioning. And so, Don, if you could go ahead and take control and just, I'm not going to be able to talk over this animation, but if you could run that now, that'd be great. Thanks so much. So we switch back to the slides. We able to do that. Okay, so when we looked a little bit more at the regulation of this receptor in Alzheimer's disease, we found the results that are shown on this slide. Here we're comparing a control receptor, which is shown in the left-hand side, both in its expression in normal as well as Alzheimer's disease patients, to expression levels of the target receptor, which is shown in panels on the right-hand side. What you're looking at is both total binding and nonspecific binding, respectively, in these tissue sections. And what you can see in the case of the control receptor is that, like many other receptors, this particular receptor is, we lose expression in Alzheimer's disease patients compared to cognitively normal individuals that are age-matched. And this particular receptor is lost as we lose neurons. As we have cell death in Alzheimer's disease, we lose expression because the cells are going away. But when you look at adjacent tissue sections from these same patients, you can see that we don't lose expression of the target receptor here. And in fact, the expression levels are really commensurate in both normal and Alzheimer's disease patients. And so what this suggested to us is the possibility that this receptor is actually upregulated on the surviving neurons and glia of these particular in Alzheimer's patients. Certainly, it's never a great thing to have the receptor that your drug is targeting changes to expression levels during the course of the disease, but at the very least, it's going in the right direction. And this is sort of supported by data from the dog studies. The human tissue studies were done in collaboration with Bob Mack and his group at WashU in Philadelphia. And the studies that I'm showing you here were done in collaboration with Rolf Craven and Liz Head at the University of Kentucky. Here, what we're looking at is receptor expression in the CSF fluid of both young and old beagle dogs, which, as many of you know, is an Alzheimer's disease model. And we see a striking upregulation of receptor expression in the aged animals compared to the young animals. So it certainly seems as though we're seeing an increase in receptor as time's going on. So putting it all together, it sort of presents a strategy for going to the clinic that is sort of matching the preclinical proof of concept with the clinical proof of concept. So preclinically, the goal would be to look at the behavioral improvements that we get in transgenic Alzheimer's models as a function of receptor occupancy that can be either measured directly with micropet by labeling our target compounds, our actual compounds, or using clinically approved radiolabeled standards for this receptor. And then matching that to a clinical proof of concept in which you dose your compound at levels that will achieve 50 and 90% receptor occupancy, and then look at the cognitive performance of the patients themselves. We're emboldened here because therapeutics that have been directed against this target receptor have actually gone as far as phase 2 clinical studies in the past. And so they seem to be, there doesn't seem to be any inherent problems associated with these particular receptors, drugging these particular receptors. However, all of the previous clinical compounds are either inactive or much less potent than our compounds, despite the ability to displace the radiolabeling at this receptor site. There seems to be something special about the atomic residues that our compounds are actually contacting, despite a commensurately high affinity at this target receptor. What we can say is that there really haven't been any, this receptor hasn't been implicated in Alzheimer's disease previously, and there really haven't been any clinical trials with therapeutics for Alzheimer's patients that are selective against this receptor. Putting it all together, we actually we believe, validated one of the only novel targets for Alzheimer's disease modification. And we did that despite the lack of clarity on the absolute structure of the ligand itself, the pathological ligand itself, which we believe is oligomers. We think that there is evidence that these species can bind saturably and reversibly to a single site, which is this receptor or something very tightly regulated by it. But the therapeutics, because of this fundamental pharmacological basis to the action of these oligomers, which we think are responsible for Alzheimer's etiology, we actually are able to produce not only prevention with these compounds, but also effective displacement of pre-bound oligomers and actual restoration of cognitive performance in the aged transgenic animal models. So this gives a really straightforward route to the clinic and a straightforward way to test this hypothesis simply as a function of receptor occupancy of our small molecules at this particular target. And we should be able to see a relatively rapid clinical proof of concept with these compounds. So that's all at this point. I think what I'll probably do is just point out to you that you can get continuing ed credits by going to the website that's listed here. And at this point, I'm going to switch over to the Q&A window and start going through the questions that have been listed here. So the first question is, do you know which other pathways in a cell are affected by these Abeta 42 oligomers? At this point, I think we've got some hypotheses that we're working on, but we're hoping to actually be able to investigate these pathways in collaboration with academic groups. I think there's a limited amount of this work that we're going to be able to do inside our company because of the focus that we're going to have on just forwarding these compounds to the clinic. There certainly are some mechanisms that we'd like to investigate in greater depth, and unfortunately at this point that's just about all I can say about what we do know. The second question is, how are the molar concentrations of the oligomers calculated? Is it based on molar equivalence of the monomer? That's correct. We're doing that is what we're doing. We're expressing the concentration as a function of monomer concentration. The actual amount that's in the oligomeric state is calculated based on monomer equivalent. The third question is, have you used human iPS-derived neurons generated from AD patients to check if your compounds prevent Abeta-triggered synaptic deficits, structural, functional? That's a very good question. We'd certainly like to do that. We have not done that at this point. We think that's very exciting technology that we would really love to check out in terms of our compound mechanism of action. The fourth question is the inhibition of your compounds mediated by prion protein, since it was proposed to be a receptor for Abeta oligomers? That's a very good question. We certainly think that work is actually really very good work. We've been able to investigate some of that in our labs. I think this is sort of the basis for us saying it. The situation is likely to be quite complex. Unfortunately, that's just about all I'm going to be able to say about the interaction of prion and the receptor that we've identified at this point. I apologize. The fifth question is, do you have any idea what mechanism triggered the amyloid and tau protein to proliferate indefinitely? I'm not sure what the author meant by proliferate indefinitely. Certainly, amyloid monomer is produced and released in an activity-dependent manner in the vicinity of the synaptic cleft it likely reaches concentrations in that vicinity that are transiently quite high. I'm not sure if that answers the question. The next question is, have you treated any one of the molecules in the aged dog dementia model? No, we haven't so far, but that's something that we'd really actually love to do. I think that that's an excellent model and certainly bears testing. There's no question. Another question is, how stable is the binding and what is the on and the off rate? That's an incredibly good question. What we're pretty excited about is the possibility that if these compounds are actually able to displace Abeta oligomer binding from the receptor, you could actually use these compounds to directly measure the off rate of the Abeta oligomers. I'm not sure how you would measure the on rate. Quantitative labeling of Abeta oligomers is quite challenging. Getting a reliable indication of a reliable quantitative measurement of the amount of label that you had attached to the oligomers and the oligomer species in the face of structural heterogeneity of the species is quite challenging. You can certainly measure the off rate of the material as we have in those ex vivo experiments. And that's something that we'd love to do in collaboration. And plans are in place to do that. I think certainly the first order of business would be to establish equilibrium conditions whereby that's happening. I think it would give a very good insight into the structural equilibrium between the various forms of oligomers, between the monomeric and oligomeric and fibrillar forms of the protein, and allow us to begin to probe the actual, by pushing on one sort of structural form, enable us to see how that actually evolves and how the structural forms of Abeta actually progress as the disease progresses. And so that's all I have for questions at this point. Thank all of you very much for listening to me. I really appreciate it. We continue to look for academic collaborators. We'd really love to hear from you, and we'd really love to hear your feedback on the evidence that we've suggested, and hope to be publishing it shortly. So thanks again very much for your attention today.